everyone and welcome back to our channel, and we are watching Pharma Galaxy. Today in this video we are going to learn about Western blotting. Imagine you are a detective trying to find out a specific suspect in a crowded city. You have a very specific description and you need a way to shift through the crowd and to find that person. In the world of molecular biology, Western blotting is a technique that acts like that detective, allowing us to pinpoint the specific proteins within the complex mixture. In today's video, we will explore this essential technique in detail. We will cover what Western blotting is, the principles behind it, the step-by-step procedures, and why it's such a crucial tool in life science, particularly in qualitative and quantitative determination of protein. Let's start with the topic. Here is the table of content which what we are going to discuss. So first one, we will be discussing about the principles, reagents required, the procedure, detection methods, and their uses. So the very first thing we need to know is what is a blot? Why we are using the term blotting here? So transferring the macromolecules from gel to membrane followed by the detection on the membrane using antibodies known as blotting. So what we are doing here, we are transferring the molecules, we are transferring the protein from that gel to a membrane. That's why it is known as blot. Okay. Molecular methods used to it is a molecular method used to identify and measure the specific DNA, RNA, or protein complex and biological mixture. So it is a technique for transferring what we are doing. We can transfer DNA, RNA, and proteins onto a carrier so that they can be separated, and then often follows the use of gel electrophoresis. So here is a small mnemonic, that is no drop. So for determination of DNA we use southern blotting. For determination of RNA we use northern blotting. This O is basically nothing, just for sake of making mnemonic, and W is for Western blot. Western blot means for determination of protein we are using Western blot. Okay, so here are various immunoblotting techniques. So Western blot is a kind of immunoblot technique. Why it is known as immunoblot? Because we are using antibodies for the sake of determination. We are giving external antibodies, and those antibodies are binding to that particular protein, and on application of certain substrate and enzyme they give certain color or something like that it is. So immunoblotting techniques uses antibodies to identify target proteins. That's why they are called immunoblotting. They involve identification of protein targets via antigen-antibody specific reactions. So as we have already discussed, southern blotting is transferring DNA. Northern blotting is associated with RNA. Western blotting is associated with protein, and Eastern blotting is associated with again protein, but it is associated with something called PTMS, that is post-translational modifications. So you're going to learn, means most of the time people will ask you, and also in certain exams also they might ask, means jiski aap preparation kar rahe ho, it might be GPAT, NIPER, anything. It's very basic, you should learn. Fingertips pe rakho. S for southern blot, S for south, S for DNA. Why DNA? D for DNA. DNA को D दक्षिण कहते हैं हिंदी में, ठीक है, दक्षिण कहते हैं, DNA को दक्षिण में कहते हैं. So you can relate like that. Northern blot RNA, Western blot protein. Very simple. So why it is there is a story. Sir Edmund Malloy Southern has invented DNA for the first time. For determination of DNA he has used the term southern blot. So for that thing, for that reason, later when for RNA something came, they termed that like northern blot. So immunoblotting techniques. Northern blot, same, just a visualization of previous slide. Next, Western blotting principle. Why Western blotting? How it is working? So Western blotting is a technique used for identification of particular protein in a mixture that we have already discussed. In this method, labeled antibody against particular protein is used to identify the desired protein so that it is a specific test. Why it is a specific test? Because for that particular protein there is a specific protein. For that particular protein there is a specific antibody you are giving. Okay. For that specific protein you are giving a specific antibody. That specific antibody is specific to that protein only. Means if there is that protein only, then only it will bind and then it will be giving some fluorescence or blot. That's why it is specific. Means if it is there, means it is there. Okay. Western blotting is also known as immunoblotting because it uses, already we have discussed. Next. Okay. So here is the procedure. The very first step is sample preparation. Here in this slide, sample preparation is not written here properly. So first what they have done, we have started, the visualization is from SDS page. This is a gel, okay, and this gel has wells, and in these wells we are going to load equal volume of protein we are going to add. Equal volume we are going to add, and by that equal volume there should be, we are also adding the equal amount also. Means per gram, if we are adding per gram 40 microgram should be there in one well. So for that calculation 40 microgram per well. So we will be calculating it, means 40 microgram. For that 40 microgram there should be equal volume of protein we are adding. So that we will be discussing later, that we are adding specific loading dye into it for making that. That is very that we will be discussing later. That is here not needed. Okay. Load and separate protein samples on SDS page. So here we will be loading this protein samples onto the gel. Next step is what will happen? Gel बनाएंगे, gel में उसको run कराएंगे. Gel will run. Okay, gel will run like this from negative to positive. Proteins are negatively charged. Okay, proteins are negatively charged proteins, they will move from negative towards positive side. ऐसे move करेंगे. तो यहाँ पे होगा क्या? What will happen here? Here the higher molecular proteins will reside here. Means one with the higher molecular weight will reside here. Those of smaller molecules, with the smaller molecules, proteins will move like this. They will move faster. Okay, how? So this gel have certain mesh. Okay, it has mesh. So when electricity is passing through it, it will pass like this. Okay, it will pass. The higher molecular, so the major principle, what principle says, that the higher molecular weight will stay on the basis of their molecular weight. On applying electricity on the basis of the molecular weight, the proteins will be separated. That you have to remember. So those with the smaller molecular weight will be residing here. Those with the higher molecular weight will stay on the upper side of the gel. So electrophoretically transfer, another step is transfer. Electrophoretically transfer fractionated proteins into PVDF membrane. So PVDF membrane, abhi we have separated the proteins in different, like we have separated proteins. We have separated proteins here. After separating proteins, what will happen? Another step is transfer. We'll be transferring this entire gel things, all the proteins which are separated in different levels. They are separated, and next step is to transfer them. So we will be transferring it on a membrane. What membrane it could be? It could be a PVDF membrane, polyvinyl difluoride, and another is nitrocellulose membrane. Okay, it is a nitrocellulose membrane we can use. But the most popular membrane is PVDF membrane we are using generally. Another step is blocking of the membrane with the neutral protein. It can be BSA is bovine serum albumin, or it can be milk, it can be skimmed milk. it will be blocking is done. Okay, after why blocking is necessary, we'll be discussing in the upcoming portion. So blocking, why we need blocking? We will block the surface. We'll block entire surface. First thing, blocking. Why blocking? We don't want any unspecific thing to bind on our surface, right? So blocking. Entirely we want to give a specific can say equal level, means we have to block the entire membrane, nothing else should be there. Block means a plane surface we need now. We need a plane surface, so for that, making that surface plane, we are going to block. We'll be blocking it with BSA, and BSA is done for around 1.5 to 2 hours sufficient. Another step is incubate the membrane with the primary antibody. So it can be overnight incubation is recommended. Overnight incubation will be allowing the surface to block with the primary specific primary antibody. प्राइमरी एंटीबॉडी फॉर दैट पर्टिकुलर प्रोटीन, टारगेट प्रोटीन के लिए एक प्राइमरी स्पेसिफिक एंटीबॉडी होगी, उस एंटीबॉडी को वो बाइंड करेगा. अनदर, incubate the membrane with HRP labeled secondary antibody specific primary, which is specific to primary, means first here is your antigen. Okay, your membrane is having antigens, your membrane will be binding, your membrane is allowed to come or to bind with the primary antibody. This is the primary antibody, your antigen is there and primary antibody will bind. After that, what we will do, we will incubate the membrane with HRP labeled secondary antibody. So HRP here is substrate. HRP labeled, HRP is what? HRP labeled secondary antibody. HRP, sorry, it's not a substrate, it's an enzyme. Okay. Horse radish peroxidase is an enzyme. It is labeled. It is labeled with the specific secondary antibody. So specific secondary body is there. When? Now it is done for approx 2 hours. After that, after this, we will be giving it a substrate. What substrate is here? For substrate, it can be ECL and peroxidase. So this steps comes during visualization when we give this substrate part, and when we will add the substrate, the images, it will give certain fluorescence, and that fluorescence is deducted. So here what we have discussed in the previous slide, that is the step by steps, the things are written. I'll provide you the PPT, or you can note it down and you can just learn it for the sake, just practice. So the three major things which we have seen, first thing is separation, another thing is transfer, and next one is detection. Separation, transfer and detection. Extraction of proteins. Extraction of proteins. How you will extract proteins? So extraction of proteins is a specific procedure you have to follow. Means there could be two things. You can it might be that protein might be from animal tissue, or that might be from cellular tissue. So if you are taking the cells from cell culture, if you are taking cells, then there is a different procedure of making lysate, protein lysate through cells, that is different. Protein lysate from a tissue, that is different. For tissue, we are using lipolysis buffer for making the things, for making the lysate, but for cellular things, for cell culture, we use PLB, that is protein lysis buffer. You can use repo buffer also, that is a specific concentration that you can use, and you can lyse it down, and after lysing you have to vortex it for half an hour at least, and all this process should be done in ice. So after using vortex for half an hour, you have to go for where? You will go for centrifuge. After centrifugation, proper centrifuge you have to do for at least 12 to 15 minutes at 12,500 RPM at 4 degrees. That is very essential, otherwise your protein is going to be degraded. So next, protein extraction part is done. After protein extraction, you have to go for gel electrophoresis. Gel electrophoresis is basically something for separation process. Means there is a gel, 3D gel, and you will be loading your samples. Samples will run by the influence of electricity. Uh, by the influence of electricity, there will be separation on the basis of molecular weight. Molecular weight means higher molecular weight will stay on the upper side, upper portion, whereas the smaller molecular weight proteins will run towards the negative end, towards the positive end, that is cathode. Next step is blocking. So blocking is very essential. You have to use for 3% to 5% BSA. BSA or milk, that is skimmed milk, you can use. Another step after blocking is addition of primary antibody. Incubation with primary antibody, specific protein which you want to see. Suppose you want to see GAP-DH, you want to see BAX, BCL, caspase, PARB, or anything. You want to see certain apoptotic proteins, you want to see if my drug or anything, if that particular protein is expressing in this particular set of protein samples, or instead of this, in this tissue or in cells, is this protein expressing or not? So what you have to do, you have to treat yourself with primary antibody treatment, which will last for approx 8 to 12 hours. That is more than enough. After that you have to go for washing. Half hour washing, in 5 minutes interval you have to wash. And how you have to wash? With what? You cannot wash it with normal water. You have to make PBST. Okay. Similarly for primary antibody also you have to make. It should be made in 3% BSA, and that BSA should be made in PBST. Then treatment with the secondary antibody. That secondary antibody should be enzyme linked. Okay. That antibody is enzyme linked. That is linked with something. Secondary antibody is linked with the certain enzyme. Next we have to treat that specific substrate. When we will be treating it, means at the time of visualization. After secondary body incubation, we have to wash it again, like we have washed earlier. We have to wash them. Why we are washing? Washing is important to remove the excess antibody. Okay, to remove the excess antibody, if it is bound somewhere non-specifically, we have to remove it down to remove the noise or to remove the unwanted dark patches on your blood. So after that there will be treatment with the specific substrate. Substrate will be ECL, alkaline phosphatase, ECL or anything like that, ECL and peroxide. Next step. So here are just basic solutions and reagents which are necessary for lysis buffer. Here are certain things. Which so here are the major ingredients of protein lysis buffer and lipolysis buffer. What should actually they consist of? So it is tris HCl with the pH 8, sodium chloride, and these are certain things. You just need to read it once. Another step is making of gel. How to make gel and how are the gel forms? What are the major components of gels? Okay, so gel is basically not a single gel is there, that a single solution you have to add and it will be formed. No, it's not like that. It is the combination of so many components. First thing, the major thing, gel is made up of polyacrylamide. So polyacrylamide gel is a cast as separating gel topped by stacking gel. So here it is combination of two gels, stacking gel and running or resolving gel. Stacking gel will hold protein here into their combs. Means here from outer side we are keeping a comb, and when we will remove these gel, these wells are formed like that. Okay, where the proteins will be loaded. So the major difference what you have to learn. First thing, pH difference between stacking gel and running gel. Another thing is 5% acrylamide. 5% acrylamide you can use here. 5% acrylamide. क्यों यहाँ पे 5% acrylamide क्यों use कर रहे हैं? Just for holding the cells. यहाँ पर क्या होगा? जो molecular size है, जो इनके बीच का जो mesh grade होगा, वो बड़ा होगा. इसकी वजह से क्या होगा? Whole amount of protein, the entire protein will pass through electricity and will come to here. Here we have higher concentration of acrylamide. Higher concentration, that is 12% acrylamide you can use, or 10% acrylamide you can use. Why? To provide the compact pores, means smaller size of pore will be provided through higher the concentration of acrylamide, smaller will be the pores. Smaller will be the pores means higher will be the resolution. Means smaller and smaller things can be separated down. So running gel is also known as resolving gel. So first thing, how you will make, when we are going to prepare gel, first you will make the running gel. Then after that, when this running gel is ready, then you will be pouring the preparation of stacking gel. Okay, so HCl for maintenance of pH. Okay, acrylamide for the making of this mesh, for casting of the gel. So stacking gel causes the protein in the sample to be concentrated into a narrow band at the top of the separating gel. Separating gel को यहाँ पे आप रखते हो, ठीक है, पूरा का पूरा set of protein यहाँ पे आता है, यहाँ पे आके stand करता है. Need to provide them a uniform, a common place. How it is going to, from where, means if you want to make someone run, you have to provide them a starting point same to everyone, right? सबका एक starting point similar रखने के लिए we are going to use this stacking gel. प्रोटीन को concentrate करके एक starting point पे लाने के लिए हम क्या use करते हैं? पॉलीक्रिलामाइड का stacking gel use करते हैं, ठीक है. Larger pores here, pores are larger. Ionic strength is lower. pH is lower, 6.8 is pH. Always remember, pH of stacking gel is 6.8. The examiner is always going to confuse you. In Viva, they are generally asking this question. pH of stacking gel, why the pH of stacking gel is different? That is a major question. You have to answer it. Running gel or resolving gel, the pH is basic, 8.8. pH is 8.8. Acrylamide could be 10% or 12% depending on the need. Here the size of pore size is smaller. Higher ionic strength is there. Investigator adjust the acrylamide concentration and manipulate gel, pores of gel as per their need. आपको बहुत छोटा size का protein चाहिए, आप और ज़्यादा concentrated acrylamide use करेंगे, ठीक है. So first step, as we have already discussed, loading. Here loading will be done. Proteins are negatively charged, always remember. They will move from negative to positive anode. Positive cathode की तरफ move करेंगे जब electricity provide करोगे आप तब, ठीक है. Another step is gel transfer. Here gel is being transferred. एक कैसे use करेंगे? Sponge, gel, PVDF membrane, again sponges, and electricity is provided. And here you have to keep it around 20 to 25 watts for 50 minutes depending on your need. Another step is antibody staining. Can be antibodies. First thing, you have to block, add primary antibody, add secondary antibody, and fluorophore, it is tagged. So when substrate comes in contact with the secondary antibody, the enzyme it is linked to, it will give certain color and you will be able to visualize it. See here, electricity you can see 75-50. So lower molecular weight protein will reside here. They will move faster. Kilodalton proteins be identified. Proteins transferred to a solid membrane. Here this is the membrane and this is gel. Proteins been transferred. After that you don't need this gel anymore. You can, if you want to check, you can go for Komasi staining of the gel. If you don't know, you don't need it. Not necessarily. If you want, you can go. Preparation of gel. Here comes the reasoning portion. So acrylamide is made up of, means how is acrylamide is there only? No. So chemical copolymerization of acrylamide monomers with cross-linking reagents are NN-methyl bisacrylamide. You have to remember, acrylamide and bisacrylamide, they will result in transparent gel. When they will meet, they will form gel. So another activators are there. There is SDS, 10% SDS we are using 10% SDS. We are using tris HCl. It is a free radical catalyzing. Another we have ammonium persulfate and timid. Ammonium persulfate, what it does, it activates timid Timid will activate this ammonium persulfate, and this ammonium persulfate will activate acrylamide and bisacrylamide copolymerization. Timid catalyzes ammonium persulfate ion to start the free radical generation. So like this, there is ammonium persulfate and timid will come in contact, it will form radicals, and after that the copolymerization will start. Okay. Next, lamellae buffer or loading dye. So we are using, for loading dye we use 2x lamellae buffer. For preparation of 2x lamellae buffer we use SDS spray. We use SDS, sodium dodecyl sulfate, 4%. 2 beta mercaptoethanol, it is toxic. Okay, so always be considerate about it, always be cautious about it. Glycerol, bromophenol, just HCl. So here are the uses you have to remember. SDS is anionic detergent. It is here, the major purpose is detergent. 2 mercaptoethanol, it breaks down the non-covalent bonds. It breaks down the non-covalent bond and make the protein linear. Okay. Glycerol is for density. Density and high resolution means you want the protein if that in the solution which you are providing in the form of wells. In the wells which you are going to load that sample, that protein sample, it should be little dense, right? It should be dense. It should not means you are pouring it and it is mixing with another solutions. So there will be nothing, means it will mix, and so we want it to be dense to settle at one place. So for that we add glycerol. Then we have bromophenol. It is tracing dye. HCl is contributed as a zeta ion effect and pH. So the next thing we have is running buffer and transfer buffer. Running buffer, basically it is having just glycine and SDS. Transfer buffer, again it is having same, just certain differences in their concentrations. Here 25 millimolar is 3, again same, glycine same, SDS there is some difference. For transfer we need 20% ethanol. 20% ethanol for proteins. Larger than 80 kd, we recommend SDS to be included at final. You don't need to just learn the components. Learn the components. Transfer is having extra, methanol is extra here. Rest everything is same like running buffer. So punctual staining, I didn't told you about it, but this is done to check that transfer was proper or not. Means suppose you have run a gel where you have loaded certain samples like 12 wells or 15 wells, 12 wells or 10 wells. You have loaded, okay, but you don't know because it is transfer, it is transparent membrane, right? And after transfer, it is transparent membrane, and while transfer you don't know if it is done properly or not. If it is done, so for validating if it is done properly or not, we can go for poncho stain. See here in poncho stain you can see that okay, it is properly done. Here the lanes are visible properly, so I can go for further detection. Means see, Western blotting is very tedious. It is easier, but it is tedious and time taking. If things are going well, it is okay. Suppose your transfer is not proper and you kept for 12 hours antibody incubation. Suppose it fails here, right? If it fails, what will happen? You will not get proper results, right? That time will be failed, means your experiment is not gonna work. So for that process, you can check if you have suspect that might be transfer is not okay. You may not go forward. So moving next. Here actually it is just the detailed way how we have to extract proteins from animal tissue or from cell tissue. So just to cell lysis, it can be mechanical centrifugation, precipitation, it is done. So that's all here for extraction of proteins. So for extraction of proteins, two things you have to remember. If you are using animal tissue, go for lipolysis buffer or acetone precipitation. Another thing, If you are using cell lysis, if you are using cells, if you want to check any expression in cells, you'll go for PLB lysis, or you can go for direct lamellar lysis as well. Next step, the same, just read it once. Sample preparations. What we have done? We have cells. Okay, we have cells. We have lysed it. We have used lysis buffer. We went for centrifugation. After centrifugation, lysis release certain proteins. What will happen? There will be some things. There will be a pallet. There will be a supernatant. You have to take the supernatant. After taking the supernatant, collect the supernatant. Collect supernatant, and that supernatant is having a good amount of protein. That's all. That is sample preparation. That is not only sample preparation. You have collected the cells. Now another step is to quantify it. Means what protein you have got, what concentration of protein per microliter, how much protein, how much microgram protein is there per microliter. That you have to calculate. There is a separate calculation you don't need to know here. For theoretical basis, just to remember cells, centrifugation, lysis will happen. You will get a pallet, you will get supernatant, collect that supernatant, that's all. So, another step, sample preparation you have done. How you have done sample preparation? You have collected that supernatant, you have collected that protein, okay, protein lysate you have. Okay, you have to add it to the loading die. You have to load it with loading die, and you have to load. Just remember the equal amount of loading die you have to add in your samples. If you have to make the samples equal. Suppose you are going to load 20 microliter, just make like that you have to add protein and your loading die. So after adding loading die, what is loading die? You have to remember 2x lamellae. 2x lamellae you have to use as loading die. So after adding after this loading, you have to apply electricity, and there will be like this. Allow the protein to move. Allow the protein to migrate. The direction of migration it is. So once it is done, it will be like this. You can go for Comasi staining of gel if you want. And after transfer, another step is immunoblotting. We have to go for transfer. This is called immunoblotting. This is called blotting. This step is called blotting actually. There is gel, and you have to, actually the picture is a little not correct. You have to keep gel. You have to keep the blotting paper नीचे blotting paper रखना है, फिर gel रखना है, क्योंकि electricity will pass from up to, ऊपर से लेके नीचे जाता है ना, ऊपर से नीचे जाता है. तो अगर हाँ चलेगा, ये भी ठीक है, गलत नहीं है ये भी, क्योंकि यहाँ पे प्रोटीन है, ठीक है, तो नीचे से ऊपर जाएगा एंड देयर विल बी ट्रांसफर. क्योंकि पहले के ज़माने में जो हम ब्लॉटिंग प्रोसेस यूज़ करते थे उसमें 1 से 2 दिन का टाइम लगता था. अभी जो हमारे पास, तो क्यों होता था ऐसा? कैपिलरी एक्शन की वजह से करते थे. नाउ what we do, now we use electricity. So electroblotting takes 40 to 45 minutes. Another step is blocking. Blocking is very important. Why we need blocking? To reduce the non-specific binding of primary protein with the wherever proteins are there, wherever protein is there, there will be no blocking. Wherever free membrane is there, it will block the free membrane area. Free membrane, not the proteins. Where protein is there, it will not block proteins. It will only block free membrane. Another step is treatment with the primary antibody. You have to add primary antibody which is specific to your desired protein. See, there are so many pool of protein is there, and you want to check that this particular protein is there or not. So for checking that particular protein, you have to block rest of the things. You will block rest things. Now what will happen? That specific protein will bind those proteins which are exposed, which are not blocked. That place only the primary antibody will block. After primary antibody, 12 hours to 14 hours, you have to go for primary antibody incubation. After primary antibody incubation, there will be secondary antibody incubation. Secondary antibody incubation will direct the primary antibody. That primary antibody, now what will happen? That primary antibody will bind with the secondary antibody, and that secondary antibody is labeled with with an enzyme. Enzyme could be anything. For example, here is HRP. HRP is very popular, that is horseradish peroxidase, HRP. After this, how we will deduct? For deduction, we need a specific substrate. For that substrate, luminol is the substrate. Luminol and in presence of luminol and hydrogen peroxide, what will happen? This luminol will be converting from ground state to excited state, and while exciting, it will generate certain light. It will generate light. That light will be captured. That light will be captured through CamDoc. You can visualize them. Another step, sorry, I wrote it later. Another step is washing. Means after primary you have to go for half hour washing, after secondary you have to go for half hour washing. Why washing? Washing will remove the unbounded antibodies from the membrane. Commonly used buffer to dilute is TBS or PBS. You can use PBS या TBS में twin डाल के use कर सकते हैं washing के लिए. प्रोटीन डिटेक्शन कैसे कर सकते हैं? प्रोटीन डिटेक्शन के लिए आप HRP is what? HRP is horseradish peroxidase, alkyne phosphatase. Luminol and ECL you can add. ECL you have to add with the hydrogen peroxide and will give certain color product. It can give, it will generate light or some chemicals, chemiluminescence, colorimetric and fluorescence, radioactive reduction, fluorescent reduction, chromogenic reduction. Through these ways you can detect if protein is there or not. Like this light will come. Okay. Next, analysis. Analysis you have to go through CamDoc. इमेजिंग कर सकते हैं, ठीक है. एक्सरे फिल्म्स में इस तरह से हम बेसिकली लैब में CamDoc यूज करते हैं, ठीक है. सो दिस हाइड्रोजन peroxide will bind with luminol. This luminol and hydrogen peroxide will bind with HRP, and it will activated luminol. This luminol will be activated, and after this activation it will give certain light, and this light will be visualized or captured through the CamDoc, and image will be taken. So here is just a demonstration that this is a protein, and suppose S1, S2, S3 are three proteins, 1, 2 and 3. What we have done, हमने इनको load किया. This is the protein ladder. Ladder is something from a reference standard, can say, where we will compare. S1 is 9. S1 is means they are moving. Suppose तीन different proteins हैं, same proteins हैं, तो इसमें प्रोटीन अवेलेबल है. हमने देखा कि ओके एक बैंड आया, वो 96 पे एक बैंड था. S2 में कोई बैंड नहीं आया, मतलब यहाँ पे ये प्रोटीन नहीं है, इट इज एब्सेंट और इट इज वेरी लेस. S3 में बैंड है, ओके, मतलब S1 and S3 are expressing this protein which is at 96 kilodalton. Means it is confirming that sample S1 is having this particular protein, this S3 is having this particular protein with the molecular weight 96 kilodalton. Okay. So here are just certain applications. What could be the applications? Major applications are to detect protein expression, diagnostic biomarkers, to confirm what are the protein levels. What could be, suppose somebody is suffering from any disease. Suppose you want to check that in cancer research, that what protein is higher in these particular cells. Or suppose you are treating someone, you have a patient sample or a tissue sample that is basically taken from a cancer tissue, and you want to check, okay, this particular protein is higher, suppose P53 is higher in the sample. Okay, so P53 is higher or P53 is lower. BRCA gene, you might know, it is associated with breast cancer. If it is higher or low, there could be certain instance associated with, okay, this is high, this is low. If it is phosphorylated, or so it is modified form of protein. So all these deductions you can make with the help of Western blottings and ELISA, certain proteins like ELISA, Western blotting and all like that. Okay, so What you need to know, that if you're a life science student, if you're associated with biology, you should know what Western blotting is. And also, if there are, you have, for passing exams, you should understand, just not for sake of knowledge, not just for sake of passing an exam, you should know them if you are the part of biology. And moving next to the disadvantage part, why, what could be, there are various possibilities that there could be some non-specific binding could occur, and it could give certain false results, poor results, as कोई ऑफ टारगेट बाइंड कर गया एंटीबॉडी में तो हो सकता है ना. Deducting and imaging the results can be expensive. That's true. If you are running a gel in your lab, it is definitely going to cost you around 4 to 5,000 rupees, because see how many reagents you have to use. It is so much long process. It takes time, it takes patience. Suppose you fail, one experiment fails, entire day. It's not only a day. 12 hours of incubation, 4 hours to 2 hours, 4 hours of blocking, 2 hours of another secondary antibody incubation, preparation of gel, and so many things. So it takes time, effort, and patience. So that is a portion, we can say, disadvantage. Multiple times you have to pipe it. Trained staff should be there. Challenges in getting a trained staff is important. In conclusion, only the important thing I have to say, that it is important, it is crucial for understanding the expression and activation post-translation modifications of these proteins involved in any study, not only in cancer progression, anyway. Any disease you are targeting, you have to run Western blot. Use a Western blotting study for potential drug candidates, you have to perform. For checking that what drug I have made, if you are a chemist, you have developed certain drug, you have to go for the cytotoxicity study. You can go for Western blotting, you can check if this expression of this particular protein is going up or down. So